We begin today with a possible breakthrough in the fight against HIV. A cure could be closer to a reality. Scientists at Temple University have successfully removed large chunks of the virus's DNA from living tissue for the first time. Researchers used gene editing technology to degrade DNA of the most common strain of the virus, HIV-1. While the tests were done on mice and rat tissue, trials on human tissue are not that far off. To discuss the new findings, I'm being joined now by Dr. Chen Liang of McGill University. His work also uses gene editing to remove HIV DNA from human cells. Doctor, thanks so much for being with us today. Your work has focused on using gene editing to attack HIV DNA in human cells. What is gene editing exactly, and how does it work?
Gene editing actually is a tool that researchers have developed to mutate the DNA sequence of a gene, as you will, in the cells. You can fix mutation, or you can delete a gene that you don't want. Yeah.
And what are the implications for the use of this technique?
It can be, in many aspects. You can fix a mutation that causes a very serious genetic disease. You can also use this technique to fight against virus infections and many other pathogens, I guess. Yeah.
Okay, so this technique can be used on different types of ailments and problems in humans. Now, in your own studies, you've tried to use gene editing to degrade the HIV virus, but the virus continued to replicate itself. Is that right? What went wrong?
What we did was that, as many other labs have been doing, we tried to use this new tool, gene editing tool, to cleave HIV DNA in the infected cells, and then the virus will be killed, theoretically. And surprisingly, what we found was that the virus can develop mutations that resist this gene editing system and then escape, meaning the virus, following some mutations, they become viable again.
Oh, I see. So they just continue to morph with different characteristics then?
Yes, yes, yeah.
Well, the new study from the researchers at Temple University shows that scientists were able to successfully use gene editing to remove DNA of HIV-1, the most common strain, from several organs of mice and rats. What do you think is new about this method?
I think this is a very important study, and it is one big step forward to apply this new gene editing tool in vivo, this time in mice. And what they showed that they can successfully deliver the gene editing system into the mice, and then this system can successfully target and cleave the integrated HIV DNA.
Do you think this is taking us a step closer to finally finding a cure?
I would say it's one step forward, but clearly there are many other, hurdles and barriers along the way that we have to overcome in the future.
Well, obviously doing experiments on rats or other animals, doing experiments on their tissue is a lot different, I'd imagine, than working on humans. What are the kinds of challenges do you think we might face in working on human subjects?
I think there are probably many, barriers we have to deal with. Humans are different from mice, and in their study from the Temple University, and their mice do not have the live HIV virus, and then the mice has the integrated virus, but is defective. And we have to be very careful sometimes because if you attack a live virus, often the virus can evolve and become resistant. And this has happened, in the early era when we used the inhibitors to treat infection in the 80s, 90s. And only until we have the cocktail therapy or the we use different inhibitors, and we can control the virus.
Doctor, I'm unfortunately running out of time, but I want to ask you one last thing. Do you think that the the reliable cure that we might someday have, do you think it exists in the world of gene editing?
I'm confident that the gene editing too will contribute to the discovery of the cure, and I think only the research in the future will tell.
Thank you so much for sharing your expertise with us, Doctor. That was Dr. Chen Liang, Associate Professor at McGill University.